Hi, I'm Christy Bielowitz with the Journal of Allergy and Clinical Immunology. I'm here at Cincinnati Children's Hospital with Drs. Gen Kano and Nives Zimmermann, authors of a manuscript published in the August 2013 issue entitled Mechanism of Siglec-8-Mediated Cell Death in IL-5 Activated Eosinophils, Role for Reactive Oxygen Species Enhanced MEK-ERK Activation. Dr. Kano, can you please summarize your findings?
We compared Siglec-8-induced cell death of primary human eosinophils in resting and IL-5 activated states. We found that while resting eosinophils die by apoptosis, IL-5 activated eosinophils undergo a process resembling regulated necrosis. This process involves intracellular accumulation of ROS and sustained activation of ERK, and this is a novel cell death pathway in eosinophils.
What is the significance of Siglec-8 for allergic diseases?
Siglec-8 is a cell surface molecule which is unique because it is expressed on all three cell types implicated in pathogenesis of allergic diseases, namely eosinophils, mast cells, and basophils. Thus, using Siglec-8 may be a good way to target these pathogenic cells without side effects of other cell types.
Dr. Zimmermann, what is the significance of your finding for eosinophil research?
The issue of how do eosinophils contribute to disease has long been studied and is still incompletely understood. Just the mere presence of eosinophils in the tissue is not sufficient to cause tissue damage, as eosinophils normally reside in the gastrointestinal tract with no ill effect. In contrast, activated eosinophils release powerful granule proteins as well as cytokines that contribute to tissue damage and inflammation. Similarly, death of eosinophils can have a protective role if it results in removal of activated cells by a non-inflammatory process, like apoptosis. However, death of eosinophils by mechanisms that release the toxic granules may lead to more tissue damage. This is where our paper comes in, showing that activation of the same molecule on the surface of eosinophils can lead to either apoptosis or a mode of cell death that's associated with disintegration of cells and release of granule proteins. Understanding the mechanism of this switch may help us intervene and steer the cells in the right direction, leading to improved disease outcomes. Importantly, inhibitors of regulated necrosis in other cells have shown significant improvement in disease processes, such as decreased neuronal cell death in stroke models and myocardial cell death in models of heart attack. Whether the biochemical pathways are similar in eosinophils and whether inhibiting eosinophil-regulated necrosis is going to improve the outcomes of eosinophil-associated diseases remains to be tested and is something we are currently pursuing.
Thank you both for your time and contribution to the journal. If you enjoyed this video and would like to see more, please subscribe to our video channel on YouTube. Thank you.